Choosing the right laboratory for peptide characterization is not a routine decision, it's a strategic one. For FDA and Health Canada submission projects, this single choice can determine whether your data moves smoothly through review or triggers months of questions and delays. Today, we're going to talk about how to make that choice wisely, not based on marketing claims or cost alone, but based on what regulators actually expect and what experienced peptide developers have learned the hard way. This is about regulatory compliance, scientific confidence, and protecting the value of your program. Let's consider a common situation. Your peptide program is moving forward with promising biology and secured funding. Samples are sent to a lab that appears qualified. But during regulatory review, questions arise about incomplete impurity profiles, non-validated methods, or reports that don't align with ICH expectations. The result is delayed approvals, higher costs, and reduced confidence in the data. This is why choosing the right lab early is critical. Federal Drug Administration and Health Canada expect more than basic analytical results. They require clear identity confirmation, comprehensive impurity profiling, and assurance of structural integrity. Analytical methods must be validated, reproducible, and aligned with ICH guidelines such as Q6A, Q2, and Q3. The key takeaway is simple. Regulators don't approve laboratories. They approve data quality and scientific justification. Peptides are not small molecules, and treating them as such is a common mistake. They have multiple charge states, complex fragmentation patterns, and are prone to degradation like oxidation and deamidation. Closely related variants can be difficult to detect without specialized expertise. A peptide-savvy lab understands these complexities and designs methods to reveal issues regulators care about. A qualified peptide characterization lab must have the right analytical tools. High-resolution LC-MS/MS is essential for identity confirmation and impurity profiling. Peptide mapping ensures sequence accuracy and structural integrity. Orthogonal purity methods provide confidence through complementary data. And related substances and degradants must be fully characterized. A key question to ask is how the lab distinguishes closely related impurities. Not every analytical method that works in the lab is acceptable to regulators. Lab functional methods may lack proper validation, scientific justification, or documentation. Regulatory-ready methods are validated to ICH standards, clearly fit for purpose, and fully documented. Strong laboratories design methods with regulatory submission in mind from the start. Regulators look beyond chromatograms. They expect clear, well-structured reports, complete method descriptions, and defined system suitability criteria. Data must follow Alcoa Plus principles, ensuring traceability, integrity, and completeness. If a regulator reads the report without explanation, it should still tell a clear and defensible story. While FDA and Health Canada are aligned, there are important differences. Health Canada often expects more detailed scientific rationale and impurity justification, along with strong batch-to-batch consistency arguments. Experienced labs know how to position data appropriately for both agencies and anticipate regulatory questions, saving valuable review time. Choosing a lab is not just a transaction, It's a partnership. The right laboratory understands your regulatory pathway, communicates clearly, flags risks early, and helps interpret results in a regulatory context. Strong communication early on is usually a good indicator of support during submission. Before selecting a lab, ask the right questions. How many peptide programs have they supported for FDA or Health Canada? Can they share regulatory success examples? How do they handle unexpected impurities? Who prepares the final reports? And do they support regulatory responses after submission? A confident, experienced lab will welcome these discussions. Ultimately, peptide characterization is about confidence. Confidence in your science, confidence in your data, and confidence that your submission will withstand regulatory scrutiny. The right laboratory doesn't just analyze your peptide, they help tell its story to regulators and ultimately to patients. Choose wisely. Your peptide deserves it.